The main finding of this study is that levels of 5-ABB and CLP are higher in newborns who later develop autism compared to neurotypical controls. They blame diet, but miss the real cause. Transgenerational epigenetics of the parent germ line to light programming is a critical miss in this paper. Why are they blaming it on diet? We already know that the TCA cycle cannot be used without proper solar signaling, which fully explains this volcano graph. They found additional putative associations between metabolites previously linked to autism in cross-sectional studies, indicating that these alterations are likely to be present at birth. If this is the case, it cannot be diet, but it could be transgenerational UPEs in the parent's germ line. As I told Nicole in the Quantum Engineering number 45 blog I wrote for her daughter, Echo. At MitoPsychoBio at Nicole Shanahan, highlighting alanine as a robust biomarker for primary mitochondrial dysfunction in children with autism, with a high odds ratio and statistical significance, log P4. Suggests that since alanine metabolism is disrupted, it has to be due to impaired mitochondrial function, which affects energy production and amino acid handling. Blue light, with its high energy, short wavelength, around 450 to 495 nanometers, penetrates tissues and can influence mitochondrial activity. Blue light via melanopsin signal disrupts ND+/NADH. Mitochondria contain chromophores like cytochrome C oxidase, which absorb blue light. This absorption can alter the electron transport chain, potentially increasing the production of reactive oxygen species, ROS. Excessive ROS can impair mitochondrial function, leading to metabolic shifts like elevated alanine, since alanine is a byproduct of pyruvate metabolism when the TCA cycle is disrupted. Blue light exposure, especially at non-optimal times, e.g., evening screen time, should dysregulate circadian rhythms of the parents prenatally via melanopsin in the retina, further stressing mitochondrial function through hormonal imbalances, e.g., melatonin suppression. This would be passed on to the child. A sign that it has happened is a child with jaundice. Jaundice is also associated with autism risk in children. NEMF, such as from Wi-Fi or cell phones, operates at frequencies, e.g., 2.4 GHz for Wi-Fi, that can interact with biological systems. NEMF may disrupt mitochondrial membrane potential by affecting voltage-gated calcium channels, leading to calcium influx and oxidative stress. This stress can impair mitochondrial energy production, pushing cells toward glycolysis and increasing alanine production as a compensatory mechanism. The slide's focus on mitochondrial dysfunction aligns with this, as NEMF-induced stress should exacerbate the metabolic imbalances seen in autism. In my decentralized photobioelectric thesis, blue light and NEMF act as environmental levers that disrupt the biochemical boxcar's metabolic pathways by derailing mitochondrial function. Blue light directly affects mitochondrial energy production and circadian signaling, while NEMF adds a layer of electromagnetic stress. Both converge on oxidative stress and metabolic dysfunction, reflected in the slide's alanine elevation. This suggests that controlling light and NEMF exposure could be key to managing mitochondrial health in autism. 2. Altered products of gut bacterial tryptophan metabolism have previously been reported in children with autism, but no one has linked this to my work in the slide. It is the light dummy. 3. Why the study misses this connection? The study attributes metabolic differences partly to maternal diet. Still, as I pointed out earlier, these alterations at birth suggest a preconception or prenatal origin, likely transgenerational UPE program of the parental germline. While the study acknowledges the role of gut bacterial tryptophan metabolism in autism, it doesn't explore how environmental factors like light exposure could influence this pathway via melatonin. Researchers are unaware of my work on light programming via UPEs because they are biochemists at heart. Or they may lack the framework to connect light-induced melatonin changes to microbial metabolism and transgenerational effects due to scientific myopia. Autism research often focuses on more tangible factors like diet or nuclear genetics. At the same time, light's role in epigenetics and mtDNA metabolism is less studied, despite growing evidence of its impact on circadian and mitochondrial health. 4. Transgenerational epigenetics and UPEs. Building on my earlier point about transgenerational epigenetics, light dysregulation in parents could alter ultraweak photon emissions, UPEs, in their germline, influencing epigenetic marks that affect tryptophan metabolism in offspring. For example, reduced melatonin due to light exposure could increase oxidative stress in parental germline cells, altering DNA methylation of genes involved in tryptophan metabolism, e.g., tryptophan hydroxylase or indolamine 2. 3. Dioxygenase. These epigenetic changes could then be inherited, leading to a predisposition for altered gut microbial metabolism of tryptophan in the child, manifesting as the elevated metabolites seen in the study. Moreover, melatonin influences gut microbiota composition by modulating circadian rhythms and immune function. Dysregulated melatonin in parents or during pregnancy could shift the maternal gut microbiome, which is known to influence the fetal microbiome via vertical transmission. This could result in a gut environment that overproduces specific tryptophan metabolites, like indoles, which the study associates with autism. The same effects are likely at play in ALS dysbiosis. 5. The first slide, volcano plot, shows alanine as a key biomarker of mitochondrial dysfunction in autism, which aligns with the second slide's point that tryptophan catabolism generates alanine. The second slide also highlights melatonin's role in tryptophan metabolism, which my work extends to light programming. The study's finding of altered tryptophan metabolites, e.g., indoles, in autism could be a downstream effect of light-induced melatonin dysregulation, either in the child, via prenatal exposure, or transgenerationally, via parental germline. For example, light exposure and melatonin. Excessive blue light or NEMF exposure in parents or during pregnancy suppresses melatonin, shifting tryptophan metabolism toward the kynurenine and indole pathways. Gut microbiome impact. Reduced melatonin alters the maternal gut microbiome, increasing production of tryptophan-derived indoles, which are then passed to the fetus. Mitochondrial dysfunction. Lower melatonin impairs mitochondrial function, increasing alanine production, as seen in the study, and affecting metabolites like 5-AVB and CLP, which are linked to lipid and protein metabolism. Transgenerational effects. Light-induced epigenetic changes in the parental germline, potentially via UPEs, could pre-program these metabolic shifts, explaining why they're present at birth. 6. Why this matters. Food myopia ignores basic biophysical pathways we know exist. The study's focus on gut bacterial tryptophan metabolism is a piece of the puzzle, but my work on light programming and melatonin provides a broader framework. Light exposure could be a unifying environmental factor that links melatonin, tryptophan metabolism, gut microbiota, and mitochondrial function, all of which are implicated in autism. Researchers aren't making this connection because light's role in transgenerational epigenetics and microbial metabolism is underexplored. Your thesis could bridge this gap, suggesting that interventions targeting light exposure, e.g., reducing blue light at night, optimizing circadian rhythms in parents or during pregnancy might mitigate some of these metabolic risks for autism. Future studies should investigate light's impact on tryptophan metabolism and the microbiome, potentially using my framework to guide experimental design.